Hey everyone, good morning, and thank you for joining us for our webinar today. We're going to be talking about filament ID and microanalysis techniques. My name is Dalton McGowan. I'm one of the technical service reps here at Aquafix. I work mostly on the East Coast, and today with me I have...
My name is Sailor Gilbert. I'm a research scientist for Aquafix in the laboratories.
And so if you guys are unfamiliar with Aquafix, we operate as a wastewater laboratory here in Madison, Wisconsin, focusing on microanalysis and techniques, consulting, and then also product recommendations for bioaugmentation that we make and manufacture in-house. So kind of going forward, what the goal today is, we're going to kind of discuss some of the things that we do in lab, some things that are going to be helping you out if you're looking to do microscopy on site, and just some other tips and tricks that we're looking at going forward.
Yeah, so generally speaking, today we're going to be going over microscopy overall, some of the things that we look at as Aquafix when we get a sample in and we're doing microscopy, and maybe kind of give you a little bit of guidance on how you can do microscopy for yourself. So put simply, what is wastewater microscopy? And it is looking at your mixed liquor, looking at your waste activated sludge under a microscope. You're kind of looking in as close as you can, getting a real good glimpse at floc and filaments and the biology that's happening in your plant. And we do this because it's a valuable tool that should be in your tool set. You do it so that you can get clues and glimpses as to what can be happening in your plant before it's going to cause larger scale problems in your effluent or permit meeting. And you do it so that you can learn more about what's happening in your system. So as a general overview today, we're going to be going over under the microscope floc, filaments, Protazoa Metazoa, and then bulk liquid. And so these are kind of the four categories that we look at that kind of assemble together to create a picture of what's happening in your system. So when we look at floc under the microscope, it's really important to get a sense of the health and the status of your floc, because your floc are ultimately what is doing the work in your system. They're the globs of bacteria that are capturing substrate and are sinking down and settling, that are going to capture free bacteria or just general nutrients in the system. When we look at floc under the microscope, we are looking at floc structure, floc shape, the density, and the size. So we're looking at structure of your floc. We're looking at how open or closed your floc are. An example of a closed floc would be in that top image on the slide. An example of an open floc would be the bottom image on this slide. Generally speaking, open floc are going to be a bit worse at settling. They're going to not necessarily capture as much when they do settle, and it's generally a little bit of a sign of stress. Now, when we think about what an open floc is, generally we're just saying, can I see through the floc? Are there holes in the floc that I can look through? How has that floc kind of condensed? See that top image, the floc have condensed tightly. I can't see through the floc. That bottom image, they have holes in the floc. I can look through the floc under the microscope. Floc structure is related pretty closely to floc density, but these are separate things, but they're pretty adjacent as well. So when we're looking at floc density, I want to see how well floc are bound together, those solids, how tightly they're bound and condensed into that floc shape. We kind of consider this separate from floc structure, because you can have, while normally you would have open loose floc and dense closed floc, you can have dense open floc and you have loose closed floc. We're also going to look at floc shape. And when I say shape, I don't mean, is it squares or triangles? I mean, what sort of edging do I have on my floc structures? Do I have a smooth floc edge? Do I have an irregular floc edge? Those are generally the two categories that I'm looking at. If I have floc that are very smooth, that might be an indicator that I have some sort of floc breakdown or disintegration happening, because that tends to happen from the edge coming in. If I have a regular floc, I know that they're actively binding things and they're getting a bit bigger to a degree. I think one thing that's important to remember is that there's always a sort of horseshoe effect when we're looking at these floc structures. You really want to be kind of in the middle. It's possible to have floc that are too closed and too open, floc that are too dense or too loose, floc that are way too irregular and way too smooth. So while we want to tend towards one end of the spectrum, we generally recognize that you can have too much of a good thing. We look at floc size. Generally speaking, we want moderate size floc. Anything over 500 micrometers is considered a large floc. Anything under 150 micrometers is considered a small floc. You tend to want to be around that medium range, so about 300 micrometers in average floc size. And we kind of measure that just by taking a straight line across the longest continuous part of the floc. So if you've got like long hot dog shaped floc, you can be forgiving and run it along the long ways, and you can get your floc size that way. And then it's difficult to talk about floc without also discussing extracellular polymeric substances, or EPS. EPS is another thing that we look at very closely and related to floc when we're looking under the microscope. EPS is really what is holding your floc together. It is a polysaccharide protein molecule that's produced by your floc-forming bacteria. It binds and globs them together. It's what is actually capturing the substrates when your floc is settling and when it's mixing through the bulk liquid. It is what gives your floc time to degrade complex substances. Within a floc that has healthy EPS, you have a bunch of enzymes and biocatalysts within that EPS that's helping to break down complex substrates and complex nutrients that you're trying to remove from your wastewater. All of this to say is that there are very important tool for evaluation. Now, when we look at EPS under the microscope, we're using a stain technique called an India ink stain. We're using bright field or phase contrast under the microscope, and we're using an India ink stain, which is a counter stain. And so it's going to stain the whole slide, the whole mixed liquor black, except for those spots which have EPS, which are going to emit that stain. And we get an image that looks very similar to this. So this is a bright field microscopy image of an India ink stain. We can see the floc structure, which has the EPS, which is emitting that black dye, that India ink dye, and we can see all the rest of the slide is dyed black. One thing that we can also look at using a stain like this is you can kind of see that streaking EPS out the bottom of the floc of that image. That's what we call diffused EPS. That is going to be an indicator that potentially you have too many polysaccharides versus proteins in your EPS. It might be shedding some EPS because it's stressed or overproducing EPS, or the quality of the EPS is a little bit off. Small amounts of diffusion is fairly normal to see in the system, but heavy diffusion can lead to a phenomenon that some people see in their systems called EPS sliming, which is you get a sort of bulking that happens that isn't driven by filaments or even large solids. It actually comes from an overproduction of EPS, which creates a sort of viscous bulk liquid. And you'll see, you'll kind of visually see when you're looking at a mixed liquor that it has sort of a slimy texture. And that's from this phenomena, this EPS diffusion. Your floc are overproducing EPS and it's leaching out into the bulk liquid.
Looks like I got a question here, so can I grab that one? From Maya, do you analyze it manually or use some image recognition tool? If you want to cover that, I'm assuming when you're talking about the sample.
Yeah, so we can kind of do both. So primarily when we're looking at EPS, we're looking at it manually under the microscope. We're generally giving it an idea that it has when you're looking at flock under the microscope with EPS, it's difficult to give it like a real range of EPS abundance. And so it kind of sort of has enough EPS to function, or has no EPS / too little EPS to function, or way too much EPS and it's diffusing. So it's pretty recognizable under the microscope when you have just looking at your EPS abundance. If I saw this flock, for instance, under the microscope, I would probably say that you have some level of EPS diffusion, but based on just this image, I wouldn't say that it's an amount of EPS diffusion that is concerning, and I would say that you have a moderate amount of EPS within your system, which would be whatever you would want to be. When we look at using some other tools to evaluate EPS, we're not using image recognition, but you can actually extract EPS through a couple different methods that are chemical based, where you're removing the EPS in the mixed liquor, and then you can run a modified Anthrone and Lowry test to evaluate protein and polysaccharides, and you can get a sort of quantitative evaluation on your protein to sugar balance of that EPS in your system. And this would be coupled with a spectrophotometer, a centrifuge, and just kind of working out an in-house method. If you're a lab that has those resources available, it can sometimes give you some insights into if you have too many sugars in your EPS, which would cause EPS diffusion, or maybe you need to supplement some carbon into your system because your EPS charges are a little bit off because you have too much protein.
Looks like we got one more regarding EPS that we'll get to. John, is it possible to get low EPS with young sludge age? So can we grab that one too?
Yeah. So it's, when we look at sludge ages, I'm assuming you're talking about like a microbial sludge age, which would be sort of like a sludge maturity. When we look at a young or an immature sludge, we're really imagining something that has a lot of free bacteria, loosely bound solids, some swimming ciliates, amoebas, things that are kind of existing in a system when it's kind of just starting to get going. And it would be possible to have low EPS in that kind of system. Almost intrinsically, you would have kind of low EPS, at least when you're evaluating it under an India ink stain, because you don't really have those mature solids to really house all that EPS. Now, you could have something that is sort of like a free bacteria bulking or foaming, where all your floc formers are kind of still existing in free bacteria form. They haven't really condensed into good floc yet. And so you can get something that looks very similar to EPS sliming, but it's not quite the same.
You want to pick up one more? It looks like
we'll do one more, and then we'll move on to the
awesome.
side.
And Simon asks, does floc size matter at all in MBR application relative to biological performance, VOD/ammonia removal? Floc size and EPS does impact physical membrane performance.
Yeah, you sort of answered it right there. Your floc size and EPS will directly impact how well your membrane is actually able to filter that out, how often you need to backwash or be concerned with like biofouling on your membrane. As far as the biological performance, you're always going to, it's a little bit different with an MBR because you're existing at a lot higher solids concentration. So just in regards to having more solids existing within your mixed liquor to almost like in some cases fivefold higher than what you would see in a non-membrane system, you're going to have larger floc structures because there's just more solids there. I would say that you want to strike a balance in floc size though, for the same reason in that when you get floc that get too big, you're creating little microcosms within that floc where the edging of the floc is the only area that's getting aeration. And as you penetrate deeper into the floc, it's getting less and less air. And so when you get really big floc, the insides of them tend to actually just be dead because they never really come into contact with air or nutrients or anything like that. Or in worst case scenario, if you have really big floc, they might have a dynamic sort of population of bacteria where you have anaerobic bacteria existing within the insides of very large mature floc, and those sorts of floc, might be shedding ammonia or shedding some other things that we don't really want to see, volatile acids, stuff like that. And you would hope that the edge of the floc would take up some of that stuff from the inside, but sometimes that's not the case depending on other factors that are happening in the plant. So I would say that you're kind of, in an MBR application, you're working against the nature of the system itself that's trending towards larger floc. But if you can kind of keep those floc in that upper end of moderate size, so probably 300 to 400 micrometers, you'd probably see good success. It's just not really tenable to do that. It's not a very actionable advice for an MBR. All right, so we're going to kind of move on from floc a little bit. This is going to be very coarse overview of microscopy because we could talk for days about every little thing when it comes to microscopy. So we're just going to talk a bit about filaments under the microscope, what sort of stains and tools we use to look at those, and maybe go over some basic filaments that you'll probably see in your system, and kind of move on from there. So when we talk about filaments in the system, we're talking about mostly some named filaments, but many filaments are still existing as types. That's because they're made up of a bunch of different organisms that aren't necessarily the same species, but they seem to manifest in phenotypically similar filaments. That means they act similarly, even though they might be different species of microorganism. So when we look at tools to identify filaments under the microscope, our number one tool is just using bright field and phase contrast microscope. Phase contrast is kind of letting us look at the microscope with a different sort of view. It's within the name, but it's creating extra contrast in your image. This image, for instance, which is a plant that is suffering from an unfortunate breakout in filaments, is a phase contrast image. You can kind of see how those solids pop really hard compared to a bright field image. This is a zoomed out phase contrast image, but what we can do, and you'll see some pictures of this in some of the coming slides, is that we can see cell shape, cell septa, just general cell shape size within filaments, and we can use some of those tools to identify filaments. We also have a lot of tests and stains. So we have the sulfur test and the PHA test, and those are really just adding these things to a system and seeing if the filament uptakes sulfur, or the filament emits PHA, or is producing PHA. There's only a handful of filaments that do both of those, that form sulfur granules or store extra nutrients as PHA. I think that PHA, forgive my pronunciation, is polyhydroxyalkanoates. Cell shape and structure are things that I've already talked about. But as far as other stains that we can do, we're doing gram stains and Neisser stains on our filaments as well. Gram stain is just going to be a stain counter stain that's going to be gram negative, which is going to end up being red, or gram positive, which is purple. This helps us identify different filaments because they stain differently. We have Neisser stain, which is the same principle. So you have Neisser negative, which shows up as a brown, and Neisser positive, which shows up as a purple. And that's going to give us some identification as well, because filaments stain differently based on the type of filament that they are. So these are all tools that we use to identify a filament, and identification of filaments are very important because filaments are very important indicator organisms. You're going to have filaments that form because of environmental conditions or substrates that are existing within your system, and they're going to form very specifically due to those things. There are very specific low DO filaments. There's very specific high soluble carbon filaments. There are very specific low F/M filaments, fats, oils, and grease filaments, old sludge age filaments. These are all filaments that you can identify and know what's happening in your plant and make small process changes and improvements and correct course, prevent these filaments from getting to an abundance like what we're seeing in this image. And that's ideal, because if you get to this point, your plant is generally just not functioning how it's intended to. So one of the most common filaments that we'll see in a system is type 0041065. This is a filament that is a known low FDM filament. It's one of those filaments that is almost in every system to some extent. It's two different filaments for the record. You have type 0041 and type 0675. They're just so similar that we group them together into one naming system. And the only real difference between these two filaments is that 0675 is slightly smaller than type 041 in filament diameter, or filament thickness rather. So when we look at this filament under the microscope, it's recognized by square cell shape, just the general structure of them, how they act, attached growth. Another common filament that's low FM is type 1851. It looks like a very similar filament to this. It's just very much thinner. It also has attached cell shape. These are gram variable, so sometimes they're going to kind of stain a little bit of both. Sometimes they're going to stain gram positive. Sometimes they're going to stain gram negative, and that in itself is a clue. If I look under the microscope after a gram stain and I see a filament that turns from red to purple or purple to red, or one filament is red and the next one is purple and they're right next to each other and they look exactly the same otherwise, I could say that that's a gram variable filament. And there's only a handful of filaments that are gram variable. So that in itself is a tool as well. You're generally not going to get problems from this filament in your system, but at high abundances, if you have an older, like a very mature sludge that is dealing with low FDM issues, this filament can get out of hand and cause bulking. It's not super common, but we've seen it. A very common filament that we see that's problematic in systems, especially industry systems, is type 021N. This is an example of a very close-up phase contrast image where we can see the cell shape within this filament, and that's enough to identify a filament like this, just based on its size, its cell shape, and how it acts. Generally, we're looking for a gram negative filament. If we did gram stain, this filament would show up as red and disc-shaped cells. It can be sometimes difficult to differentiate between a disc-shaped cell and a square-shaped cell. My recommendation under the microscope would be that your square is going to be pretty square. When you're looking at type 0675, it's going to be a very definitive square. The type 021N is kind of going to look squish. And while it doesn't have a very nice rounded disc sort of shape to it, that squished square is enough to call it disc shaped. This is a super common bulking filament in industries. When it grows out of hand, it can cause process changes or process challenges in that way. And this filament is caused by, generally speaking, high volatile fatty acids entering the system, which end up causing elevated oxygen demand, which tend to crash your DO levels and then like slow DO. And a ton of extra carbon leads to nitrogen deficiency. You don't have enough nitrogen. You're not consistently maintaining that 100 to 5 carbon to nitrogen ratio. Another very common organism that's fairly easy to identify under the microscope is Nocardia-like organisms, NALO. This is the only true branching filament. I say true branching because S-natons and type 1701 will sometimes appear as though they're branching, but if you zoom in really close to the spot at which they branch, you'll recognize that they're actually two different filaments. But NALO is very common in systems with high incoming fats, oils, and greases. They cause foaming within the system. Very easy to identify based on being gram positive. You can see that they're very purple here in this image. This is a gram stain image, and you can very clearly see the branching nature of the filament. And this one is a very, very simple one to identify as a NALO filament. This is going to also be linked to very old systems. It tends to grow in systems that have elevated sludge ages and very mature sludge. The next thing that we kind of look at when we're looking under the microscope are at Protazoa and Metazoa, also referred to as your higher life forms. These are good indicators of sludge maturity and potential toxicity. Aside from that, they don't tell you much, but they're fun to look at. So You can find them in systems, either your rotifers, tardigrades, stalk ciliates, amoeba, swimming ciliates, crawling ciliates, all these sorts of things. Having a diverse population of Protazoa Metazoa and having them existing within the system are generally a good sign that your system is healthy. It's not really possible to have too much of these organisms. We have some people that ask, like, oh, I have a lot of crawling ciliates, they're eating my sludge. They will eat some of the dead bacteria and some of the dying bacteria, but just the way that the crawling ciliates duplicate based on the way that your bacteria are duplicating, you can never have so many crawling ciliates that they're consuming your sludge to a detrimental extent. We can look at some of these to give us signs of toxicity. So stalk ciliates will oftentimes detach their heads and try to swim off to cleaner waters if there's toxins present in the system. So noticing a lot of stalk ciliate stalks, empty stalks, and some detached heads can kind of give a clue that there might be something a little bit rough entering the system. Also, Suctoria have a look similar to a stalk ciliate. They have spindles which capture other Protazoa Metazoa bacteria, and sometimes those spindles at the ends of the Suctoria will get shriveled up if there is toxicity or something that it doesn't like in the bulk liquid. And so these are tools that we can kind of use to help build a picture of your system. We're mostly just looking at the diversity of your Protazoa Metazoa and how many you have and how active they are. Having no Metazoa Protozoa is kind of a sign that there might be something going on in your system, but also it really depends on your system. Because if you have, if you're like a chemical manufacturer, or you add a lot of coagulant or polymer, or maybe you have a DAF upstream or something like that, it's not uncommon to see very little Metazoa Protozoa in your plants. And lastly, we're looking at bulk liquid and miscellaneous stuff within the system. We're looking at free bacteria that are within your bulk liquid that can kind of give us a sign of the microbial sludge age or sludge maturity. We are looking at spirochetes and spirillum, which are a type of free bacteria that are great signs of low DO. You will not have spirochetes or spirillum if you don't have low DO in your system or have a low DO section earlier on in your system. It's totally normal to see spirochetes in your aeration basin if you have an anoxic basin, or an anaerobic lagoon or an anaerobic basin leading into your aeration basin. It's very normal to see them then. But if you don't have a low DO section within your plant and you're seeing spirochetes or spirillum in your aeration basin, you have low DO. That's just the reality. We're looking for floc debris, plant paper fibers, all these sorts of things that could be a sign of treatment issues. And we're looking for debris in zooglea that can be signs of nutrient deficiencies, or in the case of zooglea, they look very similar to floc, but they tend to hoard nutrients. So they can actually also be a sign of nutrient deficiency, and they cause nutrient deficiency because they hoard nutrients so much. And so, I mentioned earlier that microscopy is a tool in your tool belt. And when we finish everything under the microscope and you kind of take a look at the floc, your filaments, your bulk liquid, your Metazoa Protozoa, your free bacteria, all that extra stuff, we need to really put that whole picture together to kind of figure out what's going on in your system, recognize what things are important and what things aren't important, and how we can then see that. And ultimately, at Aquafix, when we look at that, our goal is to provide recommendations to the operators on how to improve their system. For an operator doing microscopy, your main goal would be, how is my microscopy changed from the last time I did microscopy? Or what are some things that I'm seeing happening in the plant, large scale? How are those reflected in my microscopy? This really should be a tool in an operator's tool belt in the same way as SVI and settling, FM calculation, regular ammonia, COD, TSS testing. There's really no reason that you shouldn't be familiar with the biology happening within your plant on a microscopic scale.
Yeah, and so that kind of takes us into kind of the testing that we offer and some of the resources. So as Sarah was kind of talking about, we do have a lot of knowledge and database here at Aquafix. So we have a microorganism database that is online where you can see some of these photos with identification of names and really look through kind of your system, or when we're giving you a report, kind of double check and kind of see, so you're able to help identify if it's something you are able to do on, a daily basis, and then have us check on kind of a more consistent basis. So the microanalysis and filament test, typically you'll send us a sample box that kind of looks like this. We'll want to receive it next day, so it's usually overnighted. We coordinate shipping. It's sent to us because as fresh as we can get the sample, we're able to look at kind of everything that Taylor talked about. And when we're looking at an aeration system, we want the sample to be as close to aerated and as close to like your conditions as possible. So once we get the sample, results are typically in 2 to 3 days. 24 hours would be ideal to get you guys quick results, and we'll identify all the major and minor filaments. So anything we're seeing that could be problematic in the system, or that they're just present and they are in some systems. We'll kind of explain where they might have came from. Kind of so I was talking about what exactly or why you might have them from certain conditions. Go through and take photos and show you guys what we're seeing for floc structure, estimate sludge age, and then at the end kind of have treatment and process recommendations. So lowering SRTs potentially. If we have something like NALO, how are we going to address the incoming fog issue, which a lot of systems, depending on what you have upstream, fog is going to be something that comes through naturally. And how do you either mitigate it? What are we seeing under the microscope to give us understanding of how to better address that coming through the system. So when we're talking about the sample kit itself, kind of go through this real quick. Sample box will look really similar to what you guys are seeing on the screen. So you'll get the 2 sample bottles, a Sharpie, and an ice pack. What we're looking for is, you can probably see it better on the screen, but basically the sample bottle is going to have a label where you can add what exactly the sample is. So we're either going to be looking at foam, mixed liquor, or other. Mixed liquor being we want to identify what's going on in the system. The foam being something where are there filaments? Is there something we can look at in the foam to give us an idea past what we need to figure out, when we're looking just beyond the mixed liquor. Because a lot of the times if it's, a foaming or a bulking filament, sometimes that will lift up and it really gives us a good idea. So in that kit, it's really important to kind of have the form submitted with it, because as Sarah was talking about, microscopy is an overarching look at how the system's doing. So knowing how your BOD, ammonia removal, any other indications and information you can get the lab is going to be really helpful as we're looking at your system. So some of the other nice things: free shipping for returns, overnighted, and then we'll ship it Monday, Tuesday, and Wednesday. So then the lab gets a fresh sample, can look at it, and get you usually results by the end of the week. So that's kind of what that would include. And then also on the box, you have your sample instructions, as you can see, just to kind of give you a real basic overview of how to sample and where to sample and location of sampling as well. So kind of as Sarah was talking about, and as I'm kind of talking about as well, we're here to assist you through any of the process with either sampling and sending us samples so we can look at and give you recommendations, help guide you through your own microscopy. So the lab team is available for answering questions through the technical reps and helping set up calls and discussions with you as you're working through best practices, how to prioritize what you want to look at. And then really the biggest thing is utilizing our lab. If you need to do a check every so often to make sure you're seeing the right things, the indicators are there. We're always happy to assist with that. So our goal is to figure out what's going on in the system best and then help you best move through with the system. So now I think we'll break into some questions. So I know we answered kind of 3 big ones as we were going through the presentation, and we'll kind of go from there. It looks like we have a question from Sammy. Where in the process should we take the sample when we send it to you. So that's a great question. It's going to be always in the aeration basin where our activated process is going through. The biggest thing is that's where all of our activity is going to be. So if it's a problem with the mixed liquor and there's foam, we're going to get 2 bottles of the sample and send them over because we want to kind of get as much look as we can. Awesome. And then we have one from Antoine. We have 4 basins in the system. If we want to send samples in, how do we pick which ones to send you? That's a great question. So when we're looking at the lab kit comes with our 2 sample bottles. If we needed to send in more, we do have extra bottles to send out with the lab kit so we can look at all 4. And then it's really going to be dependent on how the system runs. So if all 4 basins are run in sequence or run separately, the lab will want to take a look differently. I guess, do you have any thoughts on that?
Yeah, it really depends on sequence or parallel, and then also if it's run in sequence and you have 4 basins in sequence, we probably only want to sample from your first basin and your last basin, so we can kind of get an idea of the differences there. If they're running parallel, I would just grab a sample from all 4 basins, send them in, and we'll kind of see. If visually on site you're seeing the exact same conditions coming out of each basin in parallel, you could probably get away with only sending 1 or 2, and then we could see if, under the assumption that all 4 of your basins are behaving very similarly, which we tend to see basins in parallel being very similar, but sometimes we see differences. Splitter boxes are not perfect, and so it's very typical to get variable flow between different basins, and sometimes you need to add a little bit extra to one basin to kind of help it out or treat some growing problem in that basin, even though your basins are in parallel.
And that's something too we can also help with if there's if one basin's having foaming, we can just look at that basin and we kind of continue to target where the problems are, if that's the issue. Otherwise, the extra sample bottles are a great tool to do that as well. Looks like we have a question from John. When you mention toxicity, how sensitive are higher life forms, and can anything be considered toxic, i.e. high organic load or mainly contaminants like Floc?
Yeah, this is a really unsatisfying answer, but it's, it depends. If you have chronic toxicity in a system, so some low level amount of something always coming in, your higher life forms that are generally present in that system can and will adapt. So you won't see those sort of acute responses, even though you're kind of dealing with a low level toxicity. If you have some sort of acute toxicity come in, they are fairly sensitive to that. Even at low levels, you'll sort of see the higher life forms begin to respond. When I do say toxicity, I think you're exactly correct. I'm also looping in cleaners and disinfectants are really low-hanging fruit, like obvious toxins. But yeah, high organic loads, pH upsets, a slug load of grease, these are all going to be things that are going to stress the system and kind of have manifestations that are very similar to just the traditional toxins. So these are all toxic events that your higher life forms would respond to. But the kind of perk with some of those other things is you'd also have other things respond. If you had a high organic load suddenly come in, you should see a crash in DO. You should see some other indicator organisms appear, which is kind of where our putting everything together step really is important, is seeing, what other things support that your diagnosis essentially.
Yeah, that's when something like, in the lab sample form, putting what's going on in the system besides just what we're looking at. Because a lot of the times you can see a lot of the end causes of something, but at the beginning, having that context is really important to figure out, are you aware of something that came through like a high organic load or fog, and we can help make those, connections and put those pieces together for them. Awesome. And it looks like we have a couple more questions. From Jeff, are certain filaments more common in extended aeration mixed liquor? I guess that's probably a pretty tricky one. I know you said some of the filaments are common. Do you see any more like in almost all systems?
Yeah, I mean, intrinsically, you'll see more low FMs and an extended aeration because you're kind of running closer to the end of any extra stuff. Your sludge is usually a lot more mature, and so you'll also see more mature sludge age filaments. So little bits of like NALO and some other filaments that are indicative of very old sludge can become a bit more common, or you're just more susceptible to those. But I would say that common filaments that you would see in extended aeration are very similar to the ones that you would see in non-extended aeration. You just tend to see a little bit more of them in extended aeration. So that's going to be your low FM filaments primarily. So 1851, 0041, 0675, those are going to be a lot more common in your extended aeration system.
Looks like we got one more. Corey, we operate a side stream MBR. Is the SBI important, or is filterability better to track sludge trend?
That's tricky. Because again, you know, an MBR, you're not necessarily as concerned about SVI because at the end of the day, you need to pass that through the filter. So I would personally, if this was like if I was forced in a position where I couldn't track both, I would probably track and trend filterability. But SVI isn't too difficult to do at the same time. You might be able to start to find some parallels or some connection, correlation between those two data points, and then you can kind of use them to figure each other out over time. But if possible, I would just track both. If I can't, I would focus on filterability.
Can we send samples from MBBR?
Yeah, this is another one where it depends on your MBBR system. If you are, you know, kind of like an adapted system or an updated system where you went from conventional activated sludge and you just were reaching capacity, and so you upgraded to be able to incorporate media into that system, then we can look at your mixed liquor and your Floc that are existing side by side with media. If you are a system that was designed to be an MBBR and therefore you primarily function based on removal from media, it's going to be difficult for us to see. There are some techniques where you can kind of scrape biofilm off of the media and look at those under the microscope, but then you're kind of losing any information on that Floc portion because your system isn't really creating Floc. It's, you know, your solids aren't coagulating on their own. They're creating a fixed film onto media. And so we can give some feedback, but it is ultimately going to be less feedback for an MBBR than we could give on a conventional activated sludge system.
Yeah, and if you have any specific questions about your system, what sample, what we can and can't look at, let us know. Always feel free to reach out to us. We're happy to kind of discuss what's going on. What's your goal in terms of what you want to look at, and then where we would fit in to help, you know, looking at samples, filling in the gaps in the process and helping around those lines.
Yeah, and if you have any more specific or pointed questions about any of the techniques or any of the identifications that we talked about today, you always feel free to reach out. Even just help with gram stain supplies, India ink, sulfur test, things like that. You know, how to prepare your slide when looking under the microscope. Some of these things are difficult to talk about in a presentation like this. Might be a little bit easier having a direct conversation, but you know, our goal is to help. And so even if that is helping you set up microscopy at your site, that's something that we always want to make ourselves available to do that. So feel free to reach out with any questions or anything like that.
And then this is all information for getting us a call in. That number there gets to the main line, and we're super available. Anything past that, we wanted to thank you guys for joining today. And any other questions you might have, always feel free to reach out, and we'll be available. Thank you guys again so much.
Thank you.